All right, now we're going to talk about toxicology. So key concepts, importance of toxicology as a tool, and how toxicity is assessed with an example. Green chemistry principle number 3, whenever practicable, synthetic methods should be designed to use and generate substances that possess little or no toxicity to human health and the environment. What we want to do in this talk is talk about the foundation for toxicity as a driver of green chemistry practices. This talk is going to be about toxicology. Terminology, I want to differentiate risk and hazard. David's already talked a little bit about precautionary principle. Risk-based decision-making is very different than precautionary-based decision-making. In the U.S., we're a risk-based decision-making society. Chemical fate, what happens to a compound when it gets in the environment, where does it move to, what happens to it on the way, when it gets there, and talk about some of the testing. So toxicology, negative effects of chemical and physical agents on living systems. Hazard, the inherent potential of a substance, so that's stereochemistry, can cause damage. And the key part can harming
Rich, excuse me, your lavalier is not, must be, the battery must be going on because we're not hearing into that.
No,
We're cutting in and out. So let's see if we can close this.
That was on. Okay, how's this one work? Okay? All right, toxicology. What I want to focus on now is hazard as the potential for a substance, given stereochemistry, to cause damage. This notion of stereochemistry and mechanism for toxicology and mechanism within chemistry is a very important linkage, because that toxicology, stereochemistry knowledge is going to be what informs design chemists to take toxicity out or find a way to work with the toxicity that may be in a molecule. Exposure, so contact between an agent and a target, and risk. Remember, I said, maybe not, you weren't able to hear, US is a risk-based decision-making framework. Europe is on their EU documents, actually call out precautionary decision-making. Very different. So risk is the probability that harm will occur under the conditions of exposure. Linking these together, so risk is a function of hazard and exposure. That means you can adjust hazard or exposure to reduce risk. And this first bullet here will show the implications of that. So with a small hazard, but with frequent or excessive exposure, you may pose as much risk as a chemical with high hazard but limited exposure. Hazard is typically what's focused on within risk reduction, at least in the past. I mean, because that takes the exposure control element out of the equation. You're going to minimize exposure. There are times where you have PPE, personal protective equipment, things like that, that has the possibility of fail. So there's a lot of emphasis on, and rightly so, hazard reduction. Okay, so let's talk a little bit about the diversity of toxicology. There's a large number of toxicology models, endpoints. You have human health, you have millions of environmental species, a myriad of test organisms, systems, assays, acute dosing, which was a single dose, higher concentration, chronic dosing, multiple dosing, lower concentrations, variety of exposure routes, a variety of toxicology endpoints. So when somebody says something is toxic, kind of scratch your head and say, okay, there's a lot of different things that the word toxic can mean. Mechanisms of action of chemicals, and I've provided some context here. Again, a complex world, a range of sensitivities. There's a couple different types of toxicology. One is this non-specific narcosis, and most organic compounds tend to have narcotic effects. Essentially, they build up in cell membranes and depress biological activity. Specific acting toxicants is what we typically think of. So there's some type of receptor binding, and that leads to some kind of a biochemical effect within the body. These are just some notations on different types of toxicology, different examples of biological systems that can be impacted. Here's a table that illustrates, for some chemicals, the intended mechanism of action, and then the toxic mode of action that resulted in a problem with that particular compound. One of the key concepts in toxicology, very old, 1493, 1541 is when Paracelsus lived, yet father of modern toxicology, is dose. Dose makes the poison. So all substances are poisons. There's none that's not. The right dose differentiates a poison from a remedy, and I'll show you some examples of that. Here you see, for a number of different compounds, the different dose required to achieve a lethal dose for 50% of the organisms. And you see a range of LD50 values across a number of different compounds. Dose-response relationships are really critical. So you have region 1, a no-effect range, a range 2 of increasing effect with increasing dose, and then a plateau of maximum effect range. There's a lot of discussion around dose-response curves. If you attend the toxicology sections, the conference over the next couple of days, you'll see a lot of discussion about low-dose effects, and I expect there'll be some charged discussion because there's a lot of information unfolding within that dose-response area. Here you can see the differentiation between a beneficial dose and a toxic dose for a number of different substances. So once again, that notion of dose making the poison. And here is from the Society of Toxicology website. You see even water. So a person died from water intoxication. Now what I want to do is identify toxicity and risk. And I'm going to talk a little bit about product testing. So this is something I did a lot of earlier in my career. And it's something that happens around the world in a number of regulatory frameworks. TSCA gets a lot of play in this country as a law that for industrial chemicals does not require toxicity testing, but more it is the obligation of EPA, for example, to have a chemical company do the testing. So there are these things called TSCA test rules that get generated. But there is a lot of testing done. It can occur at various points in a product life cycle. It has a lot of good reasons for being conducted, human health, environment, safe workplace. And these are some of the regulatory frameworks that are out there in the US, EU. You see I've divided pharmaceuticals from crop protection, from industrial chemicals. These tend to be regulated by different agencies. Generally around the world, this is the case. There's certainly exceptions, but pharmaceuticals, heavily regulated. Crop protection chemicals, heavily regulated. Industrial, big difference between what happens in the US and what happens in the EU under REACH. Now what I want to do is show you an example of the EHS evaluation for pharmaceuticals. These are probably the most well-known toxicologically molecules in the world, and I'll show you a little bit about why that is. So I'm just going to walk through these slides to give you a sense of what happens in the preclinical phase of pharmaceutical development. So preclinical is testing outside of humans before a compound is tested in humans. Then there are 3 phases of development where there is testing in humans. Phase 1 within healthy subjects, and then phase 2 and phase 3 that are conducted within patient population that's intended to use the particular drug. So here, single dose acute studies, see a variety of species that are evaluated, a number of endpoints that are looked at. Subchronic toxicity studies, repeat dose studies, again, variety of species, variety of endpoints. Chronic toxicity studies, 1 year, again, variety of species, variety of endpoints. Genetic tox studies, several genetic tox studies are done, both mutagenic as well as other types for chromosome aberrations, cytogenetics, etc. Reproductive tox studies, safety pharmacology studies, specific toxicity studies depending on the particular compound, its mechanisms of action. These studies take years and years to conduct. Carcinogenicity studies, these alone are 2-year studies, and they take millions and millions of dollars. I don't know what the latest costs on these are, but they're, I would guess, $5-10 million apiece. So there are very extensive testing done. And then in addition to these preclinical studies, there's 3 phases of clinical development. There's studies that support worker safety for the development of an occupational exposure limit, occupational exposure bands. There's process safety studies. These are the heat, pressure issues that are looked at and that Dave will talk about in Principle 12. There are environmental risk assessment studies that are required to look at environmental impact as a function of patient use following excretion when a compound may go through, for example, a wastewater treatment plant and be released into surface waters. Also in the animal health drug area, these drugs can be land applied, or they can, depending on how they're used, animals, rinsed off an animal, for example, into agricultural soil or other soils. So there's a lot of environmental testing that goes on here. And then environmental safety studies. So wastewater, for example, from manufacturing process, that's also looked at. And there's some observations and considerations here. And the first one is, I'm not going to spend any time on. This second one is where I really want to go to. So the general impressions, when you look at what the pharmaceutical industry does to evaluate safety, it's complex, it's demanding, it requires a lot of subject matter experts, it's expensive, it's lengthy, it's animal intensive, and the point to remember is it needs to be mechanistic for that knowledge to provide useful data to chemists. So this is not a process that could be undertaken for every chemical produced in industry. It's just there's not enough money, there's just not enough profit margin in the compounds that are produced by industry typically to be able to do these. So we need to find a different way than these types of long-term tiered testing to evaluate chemicals. Okay, environmental risk assessment. I want to talk a little bit about environmental transport and fate. Where a chemical ends up in the environment depends a lot on what its introduction scenario is. So if it's a spill into a water body or whatever, it's a very different framework than if a chemical is discharged through a wastewater treatment plant pipe, for example, or land runoff. So those are very important considerations for how a scientist makes a prediction about where a chemical is going to end up and what type of toxicological impacts it might have when it gets there. So these are the scientific data points that are used in making those evaluations. Environmental transport is controlled by pChem properties, things like solubility, water, lipids, volatility, charge. Most surfaces in the environment are negatively charged, which does something to control whether you're cation, anion. Environmental fate, degradation processes, not just biodegradation, but chemical and photodegradation as well. So environmental risk assessment, and there's a parallel process for human health risk assessment, is a process for estimating and characterizing the likelihood of adverse effects to the non-human environment. It can be done predictively, retrospectively. There is a process that's used, and I apologize because some of the term hazard is used here a little bit more broadly than what I defined it at the beginning of this talk, but there are different major phases. The first phase is to understand your molecule. You need to know something about its pChem properties, something about the release scenario, something about what lives in the environment where that release scenario is, because when you do your effects evaluation and you look at the hazards, that is typically done on species that are relevant to the environment in which this chemical is going to be in. So for example, if you have a release to an aquatic environment, typically the testing you've done is at 3 trophic levels, which means 3 feeding levels. So you look at an algal species, small zooplankton that feeds on the algae, and then a fish that feeds on the zooplankton. So it's a bracketed set of organisms that's tested to try and come up with an estimation about the toxicity potential and the risk potential. Hazard evaluation, talked a little bit about this. Evaluation of the molecule, you need to know something about the properties. Collection of data, there are batteries of not just effects studies that I've already described, but also fate studies, biodegradation studies, for example, to try and determine the fate of a compound, sorption studies. In domestic wastewater typically passes through a wastewater treatment plant. What that is a large bioreactor, essentially a big tank of water that has got a lot of biomass in it, and the wastewater goes into this. There's about 12, 24 hours, depending on particular plant, where the biomass in there has a chance to eat whatever is in there, removes the easily degradable material. Anything that's not readily degraded typically passes out of a wastewater treatment plant, is released into surface water, and then travels down a river to where the next intake pipe is that goes into a drinking water production facility in many cases. So that happens all around the country and the world. I don't know how many, somebody estimated how many times water may pass through somebody's kidneys on the way down the Mississippi River. But that's something that happens. A lot of water reuse out there. Okay, just talked about this. All right, the exposure assessment. So the way this works is you come up with a predicted environmental concentration using this formula. And actually, this works pretty well. We can predict, with a pharmaceutical, you know something about the volume, or any chemical, you know something about the volume that's going to be used, you know something about the amount of surface water that's out there, and you can predict dilution rates, and you can come up with pretty reasonable estimates about the concentration that's going to be out there in the environment. Then the effects assessment is done. I've already described these types of tests. And then the risk characterization. Basically what this is, start with this value called a PNEC, predicted no effect concentration, and you calculate that with your effect data, and you apply an application factor. And basically what this is the less confidence you have about your toxicity value. So if you only have one acute toxicity study, for example, you use a larger application. If you've actually got field monitoring studies where you've looked at dosing and organisms in the environment, you have a lot more confidence about how toxic this material might be, and consequently use a smaller factor. And then the risk characterization. And the way this works is you divide your predicted effect concentration with your no effect concentration, predicted environmental concentration with your predicted no effect concentration. If it's 1 or greater, depending on the regulatory framework, you may have to go back and do additional studies. There's some kind of mitigation that's typically required. And the course of action could be one of several different routes. You could try and refine either the environmental concentration or the no effect concentration to build a higher level of confidence around the number you're using. And then when you come to the app, there's a risk-benefit evaluation piece that's done. And once again, there is a human health parallel that's used for industrial chemicals and actually for pharmaceuticals as well. The pharmaceuticals are different in that for patient use, they built this ring fence around them. So that's all the language you hear when you hear direct-to-consumer ads on TV or you read where when you listen to it, that's what they're doing. They're defining who's the patient population, who takes this, who doesn't take this, and that's what's defining. And then they describe the off-target effects that people who are taking the medication should be watching for. So those are all very carefully worded and very regulated messages going out. So in summary, toxicology provides hazard data that can be leveraged to inform green chemistry initiatives, especially mechanistic toxicology information. Descriptive toxicology information is really not of much value to a chemist who's saying, how do I design my molecule? What do I build in? What do I leave out? They need to know specific characteristics of the molecules so they know what to design for and what to design out. Hazard determination and risk evaluation are broadly utilized in industry for product development and for approval. There are certainly differences between pharmaceuticals, biocides of various types, crop protection chemicals, and industrial chemicals. And as we mentioned, there's differences around the world in all of these frameworks. Trade-offs are an inevitability, and people need to understand, again, the exposure framework, the release framework, the nature of the chemicals to be able to make the most effective trade-off decisions. And as we'll talk about more in the next presentation, mechanistic toxicology and read across the chemistry is really essential. And innovation and collaboration, again, are really the foundation, and I'll go back to make this point again, that it's really all the way through the organization that happens. So thank you very much. Questions? Okay.
There's a break, and
Okay.
I suspect in looking, it's a great time to take a break because I can see the energy level is being depleted. So get up, get the blood moving again.
I hope I wasn't entirely responsible for that.
One final comment is, again, that you're going to, as I said in the beginning, you're going to be challenged across a lot of disciplines, and I'm sure that toxicology was not one of the things that you've been taught in your undergraduate education in chemistry. And again, each one of these principles could be semesters or entire course of studies through a PhD. So just keep that in mind. And 15 minutes, and we'll see you back.